Good morning. My name's Elizabeth Budde. I'm the hematologist and also the executive medical director for CETA Hope's immune effector cell program. Thank you so much for coming here, especially those who are not on site, to support our effort and our outreach. I just want to say that welcome. And if those of you who are on Twitter or X, I already tweeted, the great kickoff from Leo, and also what I learned, from about CIRM's history, the training, and its commitment to support our research and outreach. Okay, great. So today I'm going to spend the next probably 20 or 30 minutes time just to talk about immune effector cells and our CETA Hope's IEC program, how we do it. So I figured that it's probably easier if we together walk back to history, looking at the development of adoptive immune cell therapy, which really began back in the 1950s. That was even before we know what B cells are, what T cells are. There was this mouse study, demonstrated that you can transfer anti-tumor immunity by transfer those cells from a mouse to another one. So it was around 10 years later, finally people were able to identify B and T cells, and subsequently, we learned a great deal about how T cell function. We isolate, the T cells factors such as IL-2, interferon gamma, and also more importantly, back in 1980s, that was the first time, we actually learned how to effectively do gene transfer. T cells are what used to known to be very difficult to manipulate. So the gene transfer technology using viral-based approach has really greatly improved the efficacy of how we manipulate T cells. And it was also during the 1980s that this concept. This was after we learned what a monoclonal antibody looked like and how to make monoclonal antibody. So back in 1980s, that was the first concept. What if we make a chimeric receptor has both the immunoglobulins activity, but also has the effector cells function? So that was the initial CAR concept at the end of 1980s. And subsequently, we learned more about how to make immune effector cells, like these, viral-specific T cells and tumor-infiltrated T cells to expand in. And that was also when the first generation of chimeric antigen receptor was made, which was a very simple concept, a binder to a target on the surface of T cells, and which the signaling then triggered the CD3 signaling. And subsequently, with a lot of advances in our technology and our understanding how T cells function, also the methodology, how to make these adoptive T cells in vitro, the clinical trial began the testing CD19 and CD20 back in early 2000s. The first CD20 CAR T clinical trial was actually conducted at CETA Hope. And of course, nowadays, the CAR T cell look a lot more advanced than what it used to be. So it's a second generation, third generation, and eventually 2017, the first gene therapy and the first immunotherapy was approved by the FDA. That's T-cell janglusol for leukemia and axi-cell for lymphoma. And just a few weeks ago, as most of us know, that the FDA actually approved the first TIL therapy for melanoma. So the immune effector cells can have several different types. The most common one used in the clinic nowadays is really called CAR T cells. So you can see my pointer. Oh. But the top one is the CAR T cell, which has each T cells have different barcode by the T cell receptor. So what CAR T cell, the advantage of CAR T is really it can gene transfer, modify individual or ordinary T cells and change them into tumor-specific T cells by putting this chimeric antigen receptor on top of the T cells. And then they're able to recognize tumor cells and execute their killing activity. And for NK CAR, which is simply by putting the same CAR inside into T cells, but put those CARs into the NK cells, then it becomes the NK CAR. And for TIL therapy, which is different than CAR-based immune effector cells. TIL therapy are really, there's a way, those T cells are pre-existent in the tumor. So there's a way you can use a peptide that can stimulate the tumor-specific T cells and find a way to harvest these T cells and expand them to sufficient number. And those are tumor-specific T cells. So the goal of adoptive T cell or NK cell therapy is really to try to rescue and reprogram those immune effector cells to redirect them to be able to robustly kill off the tumor and at the same time also clone themselves. So basically, you can these very ordinary immune effector cells who are usually suppressed in the body, and that gets a chance to be activated and then recognize the tumor cells and knock out the tumor cells at the same time. It's a live drug because these are cells. They can expand up to at least 1,000-fold. So it's a very potential, very powerful tool that we can use now to treat tumor. And here is a very common process of how we do CAR T these days. Conceptually, so basically you have a patient. In most cases, we isolate the immune effector cell from the patient and send them to manufacturing. And those cells will undergo training to become a CAR T cells or tumor-specific T cells. And then there's an expansion process. Once the cells meet a certain dose level, if they meet all the release criteria, it can be put into as in a vial as a drug and give it to the patients. And there are many variables in this process can really affect the CAR's activities and toxicities. How to make these T cells, what kind of cocktail cytokine you put in culture these T cells, and what's the starting population and the length of manufacturing. And so many different tweaks here, really leads to different characteristics of different CAR T products. But here's a more realistic look in the clinic. Basically, you start with patient referral, consultation, and payer approval, and then manufacturing start, leukapheresis, and which also requires line placement. And eventually, the patient will be waiting. A lot of them will actually need some sort of treatment to stabilize their disease. When the cells are ready, the treatment begins. So it requires a lot of coordination, collaboration, and communication. And here's another deeper look. This is really looking at all the potential parameters can really impact the treatment, this treatment. It could be what about the capacity of the hospital bed? What about what about caregiver? Do we have a caregiver for those patients? Are we able to bring such treatment to patients who are at a slight socioeconomic disadvantage? And last but not the least, we all know that previously there was shortage of tocilizumab, and there's also a shortage of fludarabine, which severely impaired our ability to give CAR Ts. Now, of course, subsequently, people figure out a way to overcome that. But CAR T cells as a mature valid platform was really demonstrated by these initial CAR T cell clinical trials. So on the left is the historical outcome of patients, the lymphoma patient who had primary refractory disease to treatment. In these patients, looking at the results from post studies, for this patient to get to a CR, the chance was only 7%. And the median overall survival was only 6 months. And on the right are those initial patients who enrolled on ZOMA-1 using the axi-cel Kai product. Those patients being followed longitudinally. So with the 5-year follow-up, the overall survival is around 64%, so much higher than the historical control. And no new safety concern was identified. And also all the patients that have sample available have their B-cell recover at year 4. So the CAR T cell treatment has actually made another leap forward for lymphoma. Now it's become the second-line treatment. So for around 20 or 30 years, when patients have relapsed lymphoma, the standard care is really to give them a second-line salvage chemotherapy treatment, and those who responded to the treatment will go to autologous stem cell transplant. So back in 2022, 3 phase 3 studies had a readout. 2 out of 3, ZOMA-7 and Transform, actually were positive. The comparator is the salvage chemo followed by autologous stem cell transplant. So CAR T, CD19 CAR T in this setting, is the winner. So here is the recent update on the longer-term follow-up. Those patients who were randomized to receiving their own autologous CD19 CAR T versus those who were randomized to chemo plus autologous stem cell transplant. The orange line on the top is this survival curve of those patients who receive CAR T, and the blue ones are those who received the standard care at the time. You can see the curve actually separated out pretty early on and continues to remain separated. So if you look at the median progression-free survival, really favoring patients who receive CAR T of a 14.7 months versus those who get chemo plus auto transplant, the median progression-free survival was only 3.7 months. And this study actually led to the approval using CAR T in the second line for those early relapse aggressive lymphoma patients. And here just two patient cases that were treated at City Hall to just showcase how powerful CAR T could be. On the left is these are the pair panels. On the left is the pre-treatment baseline PET scan. Day 40, you can see all the signals were gone. And the second case on the right-hand side is a patient, literally the patient had around at least 10 different treatment and continued to have bulky ulcerated masses in the axilla and also inside the abdomen. And the patient was actually really was on his way to hospice. The patient was only 48 years old, but we really did not have a better solution. And fortunately, the case was actually brought to our weekly CCIC meeting where we discussed CAR T patients' care and also CAR T patients' eligibility. And this is the patient who had very poor performance status, but otherwise young and had good organ function. So then our committee also was able to convince the investigator and convince the treatment physician to say, we really don't have much more to lose, and looks like the patient's poor performance status is really because of the lymphoma. So let's give him a chance. So we gave him axi-cel, and this patient, actually went into a complete remission and continues to do well. That's 3 years out already. And so taking a step back, looking at globally, CAR T-cell therapies, clinical trials are very robust these days. There were around 500. So this is, we go to clinicaltrial.gov to search for CAR T. In 2018, when I did the search, I put out around 500 trials. But when I searched it just a few days ago, it's already 1,685 trials done globally, mostly in Asia and in North America. So there are a lot of interest in applying CAR Ts or using novel CAR Ts for different disease entities. It also has expanded to autoimmune disease. So at City of Hope, I'm sure it's the same as all the other sites. No one, not a single person can offer CAR T on his or her own. It's really a great team effort. You will have a lot of experts and nurses and supporting staff such as pharmacists and quality people, biostat, and we have dedicated inpatient and outpatient services for these patients to really make sure the best outcome of this patient. And we started beginning to do immune effector cell treatment on clinical trial back in 2012. So there were 3 such trials in 2012, and one of them is the CD20 CAR T clinical trial. And subsequently, you can see the expansion of not just the commercial CAR T, which began in 2017, and also clinical trial CAR T patients. So we treated more than around 1,400 patients with immune effector cells today. And there are 3 major immune effector cell categories these patients can fall into. One is the investigator-initiated clinical trial using immune effector cell product that developed and made on campus at City of Hope. And in the middle is the bucket where we partner with industry to conduct clinical trials using various CAR Ts or other immune effector cells for different patient population. On the right is the number of patients is the commercial CAR Ts. And the approved commercial CAR Ts, the list is actually expanding. And here, there are 6 different CAR Ts and targeting at least 10 different indications. And just this year alone, 2024, 3 more new indications are approved by the FDA. And one of them is using lysocell, there's a typo here, using lysocell for chronic lymphocytic leukemia. So that's a new disease, a new indication. And ide-cel and cilta-cel both are BCMA myeloma cells, they were actually approved for earlier line use, especially the cilta-cel now is available as a second line use. And here's a table that breaks down all the commercial product patients that were treated at CTL Hope from 2017 to end of last year. Totally, we have treated 662 patients receiving commercial CAR-Ts, and they received from lymphoma to leukemia and to multiple myeloma. And one of the major undertake that we did was in March 2022, we started to do outpatient CAR-T. You can see all the blue bar before March 2022. All the patients at CTL Hope received their CAR-T infusion in the inpatient setting. And so as a mission in March 2022, we began to do outpatient CAR-T. You can see the blue line actually gradually goes down, and the orange bar, which represent the total outpatient CAR-T treated volume. The green one is the one that patient who received both lymphodepletion and T-cell infusion in the outpatient setting. You can see the green bar continue to go up. So we are really transitioning from inpatient to an outpatient default program. And here's another look at 2023. From January to December, you can see the patient who received the inpatient CAR-T numbers it actually stays quite low. Using December, that's the last one here as an example, only one patient received commercial CAR-T in the inpatient setting, and 12 out of 12 patients received the outpatient setting lymphodepletion and CAR-T infusion. So switching to our clinical trial capacity, half of our immune effector cell patients receive commercial product, and the other half of them actually they come here, they receive they receive immune effector cell treatments on clinical trials, and we have this expanding portfolio of IEC trials for both solid tumors and hematologic malignancies, as well as for patients with infection. And recently, we just did our first case, a patient using a CD6 T regulatory CAR that developed at CTL Hope for a patient with graft versus host disease. And in this pie graph, you see a quarter of these clinical trials were actually originally and started at CTL Hope using CTL Hope's own novel CAR-T product. And this is look at the immune effector cell clinical trials that back in 2022, from 1 to 2019, we had 9. But from 2019 to 2024, in the 5 years, the number actually doubled from 9 to 18. And a lot of these clinical trials would not be possible without the funding support, including CIRM. So we received 3 CIRM grants in 2023, really a showcase of the strong collaboration between CTL Hope and the CIRM. And I'm actually not going to go through this. I know Dr. Budde, who is going to give us a keynote speech later, he's going to talk to you guys about our brain tumor program. But this is really the first case of patient ever with GBM that achieved a remission using CTL Hope's own CAR-T product. And this is BAFR targeting CAR-T is another example of the science that developed on campus at CTL Hope from Dr. Larry Kwok's laboratory, and where his group proposed that targeting BAFR would deliver and at least similarly, or maybe there's a chance to superior than CD19 targeting. This is really based on the notion that BAFR is critically important for B-cell development and also important for the survival of the B malignant cells. So hence, his group that developed this humanized BAFR CAR-T, and this CAR has a 4-1BB as co-stimulated domain, so suppose it will last a bit longer. And we open up a clinical trial using the BAFR product, manufactured, produced at CTL Hope on campus here. Now, there are 3 dose levels. So far we have completed dose level 1, and dose level 2 is ongoing. And dose level 1 is 50 million total CAR. And to our pleasant surprise, that all 3 patients enrolled and treated at dose level 1 who received 50 million CAR-T in total, all went into a complete remission. And these 3 patients, they were not easy because 2 of them actually had prior CAR T-cell treatment. And the 3rd patient has T-cell histiocyte-rich B-cell lymphoma, which historically does not respond well to CD19 CAR T-cell treatment. And this patient also had autologous stem cell transplant with relapse disease. So all 3 patients went into complete remission, and all of them continue to stay in remission. So we're very encouraged by this observation. So dose level 2 is ongoing so far. And we have treated 2 patients. We need another patient. Now, there is a possibility that we might not need to go up to dose level 3. We might just stay at dose level 2 and begin to do expansion. So last but not the least, just to give you an example of the off-the-shelf allogeneic CAR Ts, and this is a partnership with industry. So CTX130 is an allogeneic CRISPR gene-edited anti-CD70 CAR. So basically, the T cells are from healthy donor. Those donors have to meet a certain criteria. Once those T cells were isolated from the donor, and these T cells undergo multiple modifications to really get rid of their T cell receptor, so they won't be able to recognize the recipient cells. There's also some other manipulations to further make sure that these modified T cell will not mediate the graft versus host disease. And the CD70 CAR were then introduced into these T cells. So these T cells were, again, manufactured from healthy donor, and it's already available before patient get onto the study. And this CTX130 was tested, has been undergoing testing in 2 different cohorts. One set is for patients with T-cell lymphoma. A significant portion of those patients has CD70 expressed, and we know that T-cell lymphoma typically is very difficult to treat. And you don't really need to remember each individual clinical trials. For T-cell lymphoma, the overall survival is overall response rate is always somewhere around 30%, and the response rate doesn't last that long. So this CAR, when you give into patient with T-cell lymphoma in relapsed refractory setting, in a higher dose level, dose level 3, dose level 4, you see there are patients, 3 patients has CR, 4 patients that have partial remissions. This makes the overall response rate 70%. And patients with both PTCL and CTCL benefited from this study. And the second cohort of this study is a phase 1 study, actually for clear cell renal cell carcinoma. And what I'm showing here is the different dose levels and for the phase 1 portion. And Monty Paul, one of a solid tumor investigator who actually led the study, and this outcome of the study was just published a week ago in Cancer Discovery. You can see for clear cell renal cell carcinoma, using this off-the-shelf allogeneic CAR Ts can lead to, in the first patient had very durable remission, the patient being 3 years old, still in remission. And there are multiple patients who has stable disease stabilized. So disease control rate here is 81%. And this CAR right now is undergoing another iteration to really further improve the CAR persistence and potency. So I'm just going to conclude here that we know that adoptive immune effector cell therapy has really changed the outcome of patients with at least hematologic malignancies, with multiple products already approved by the FDA available to patients. But to have a successful delivery of CAR T cells or other immune effector cell therapy safely, effectively, it really requires a highly specialized clinical expertise and also a committed sustainable infrastructure support. And there are many ongoing efforts looking at how to further improve the efficacy, reduce toxicity, not just the physical toxicity, but also financial toxicities, understand the resistant mechanism from the non-responders, and how do we reduce costs, and how we expand this technology applied to more disease entities, and also how do we bring this innovative treatment to more patients. This effort is still ongoing. And thank you for your attention.